Good afternoon, everybody. My name is Jiwan Oh, and I am a neurologist at St. Michael's Hospital at the University of Toronto. I'd like to first start by welcoming you to our inaugural CanProCo patient education event. Um, I will be starting the activities today by giving you a general overview about CanProCo, which many of you are very familiar with because you have chosen to participate, which we're very grateful for. And then you'll be hearing from many of the scientific pillar leads of CanProCo about updates with progress that we've made in many different interesting sub-studies. So I'm just going to take a second to share my slides. All right. So I should note that we were very much hoping to host this event in person, but because of obvious circumstances related to the pandemic, we haven't been able to. But hope that today's various sessions that we've prepared will be very informative for you, and we'll look forward to future years where we plan to host annual events where hopefully there will be an opportunity for everybody to get together in the same room. All right, so I'll take the next 20 minutes or so to give you an overview about CanProCo. And again, I know that many of you are familiar with different aspects of the study. But hopefully this will be a nice overview, and then I'll give you some brief updates about the study itself, and then won't get into as much detail about different updates within each scientific pillar, because that's what our scientific pillar leads will be taking you through, and then there will be time for questions at the end. This is a slide outlining my disclosures, and the ones relevant for this talk are that I have received research funding as the national lead of CanProCo from the MS Society of Canada, Brain Canada, and our industry partners, Biogen, as well as Roche. All right, so as people living with MS, you're all very familiar with clinical care in MS. And you're also familiar with the fact that likely you've noticed that MS is in the news a lot, and MS care has really changed dramatically in the past decade. And amongst neurological diseases, this is pretty unique because it's not often that there's such rapid progress in treatment and in the scientific understanding of a chronic neurological disease, because most neurological diseases are incredibly complicated. And so we're very fortunate in the MS field that there's been huge developments in the science as well as in the treatment philosophy of caring for people living with MS. So first of all, because of many, many different treatments that have been approved in the last 25 years, and as you can see in the last 10 years, there's really been an explosion of the number of new treatments available. There's been a dramatic change in terms of the general treatment philosophy of people living with MS, where we are much more aggressive than we were, say, 10, 15 years ago. We emphasize early personalized treatment, rapidly optimizing treatment if a specific treatment is not working as well as it should. We use MRI routinely as both a diagnostic and disease monitoring tool, and also from a research standpoint, as we'll highlight in CanProCo. And what's being increasingly recognized is the importance of multidisciplinary care. So it's not just the neurologist that needs to be involved in caring for people with MS. It's a whole team of healthcare professionals, whether it's neuropsychiatrists, physiatrists, occupational therapists, physiotherapists, social workers, speech language pathologists, and the list goes on. So despite all of this progress, and I think it's important to emphasize the positive, it's very clear that there are many unmet needs still in the MS field. And one of these unmet needs is a better understanding of progression in MS. And obviously, this is a highly relevant concept in the progressive subtypes of MS, like primary progressive MS and secondary progressive MS. But what we're understanding more and more is that, first of all, our classification of MS probably needs a relook and some refinement. But it's not just in progressive subtypes of MS, but also in very early relapsing forms of MS, that progression can be a component, although it's very minor in many situations. So it's very clear that this is something that is relevant across the spectrum of MS, and we really need a better understanding because from a clinical perspective, there are many challenges when we're trying to deal with progression that can be seen across the spectrum of MS. So first of all, when it comes to diagnosing people with progressive MS or progression itself in MS, it can be a challenge. In terms of monitoring progression, we don't have yet excellent clinical tools. In the clinic, as you know, when you see your neurologist, we usually do neurological exams, but this is far from perfect, and clearly we need better tools. In addition, when it comes to treatments specifically for progressive MS or for this progressive component of MS, we don't have excellent treatments. And it's very important that in the last 3 years, for the first time, there have been treatments approved specifically for progressive forms of MS. However, these treatments, while it's important that they are clearly effective, they're modestly beneficial at best. And it's very apparent that we need better ways to treat progression in MS. And specifically, we need agents that will be able to repair existing damage rather than just slowing down damage that is happening. And then from a clinical perspective, it's very apparent that particularly for progressive forms of MS and progression in MS, we need more access to comprehensive interdisciplinary care pathways. So these are the challenges that we have in clinical practice. And this is exactly where the Canadian prospective cohort study to understand progression in MS, or CanProCo, we hope will play a major role in the field. So CanProCo was specifically designed with the mission of trying to establish a team of investigators to design a comprehensive study of people living with MS in Canada, specifically to answer important questions related to progression in MS. And this is because in the field, we recognize that this is truly probably the greatest unmet need. From the very beginning, there were specific things that the CanProCo study had to meet, including answering questions in 3 research domains, and these include understanding mechanisms of progression, treatments and real-world evidence, and the impact of these treatments on progression. And then we were required to use methods from 3 scientific pillars, namely epidemiology and health outcomes, neuroimaging, and neuroimmunology, and incorporate all of this into the study design of CanProCo, again, with the overall objective of better understanding why progression occurs in MS. So this was an effort that was a long time coming and involved many different funding agencies and clinicians and scientists from all around Canada. So this is a summary of the timeline of what has happened thus far. So because for many years, there was a recognition that, again, understanding progression in MS is really one of the most important things in the MS field from both a clinical and scientific perspective, it took many years for the MS Society to collect a range of partners that were interested in funding a huge effort like this. And so this was done over many years with the efforts of many different people within and outside of the MS Society. And then in February of 2017, there was a call for applications for a planning grant where within Canada, basically the call was for a group of investigators to come together that met all of the criteria that I outlined in the prior slide, where we were supposed to design a study that would best allow us to understand why progression happens in MS, but incorporating many different scientific fields of study, which, as I outlined earlier, include epidemiology, health outcomes, neuroimaging, and neuroimmunology. And so a big group of us came together, and you'll be seeing photos of the key group of CanProCo investigators and hearing from many of them later today. But we got together in July of 2017 when we were awarded this planning grant. And over a period of 10 months, met many times, had many conference calls, many heated discussions about what the best study design would be, to try to design a cohort study across Canada that would allow us to understand progression in MS. In May of 2018, we submitted a full grant to an international panel of reviewers, and the grant was over 1,000 pages long. In June of 2018, the grant underwent a review with an international panel of experts who had a wide range of experience in different cohort studies in their own countries, and also a wide range of scientific expertise. We were very fortunate because in July of 2018, our CanProCo key group of investigators were awarded the initial grant, which was in the amount of 6.6 million that would allow us to conduct a cohort study over 5 years. In October of 2018, another industry partner came in and contributed 2.12 million. And then in the spring of 2019 was when our cohort officially launched and first patients were enrolled across Canada. And then in December of 2019, the government of Alberta pledged another 1 million towards additional CanProCo investigations that would take place within Alberta itself. So that is the timeline of events that happened leading up to the initiation of CanProCo. Just wanted to summarize, who the key group of CanProCo investigators are. So I am the national lead of this study. And so the central site is St. Michael's Hospital at the University of Toronto. But we have many investigators who essentially are taking charge of the different scientific fields of study within CanProCo, and they're summarized here. For neuroimaging, we have Dr. Shannon Coland and Roger Tam, who are leading the imaging effort. In neuroimmunology, we have Dr. Alex Pratt from the University of Montreal, leading the effort. And then for epidemiology and health outcomes, we have Dr. Scott Patton, Tony Trabulsi, as well as Larry Lind leading these efforts. And as you can see, the diagram on the right just summarizes kind of the leadership structure of CanProCo, and you'll be hearing from many of our scientific pillar leads today. This is a table summarizing all of our various co-investigators, because there are 5 CanProCo sites across the country, and these represent the largest MS clinics and academic centers around the country. And as you can see at each site, because a lot of data collection takes place, including clinical data, imaging data, and biological data, we need a huge group of co-investigators with expertise in these very distinct scientific fields to enable data collection. So as you can see, a large number of people involved from all around the country. And then we have another really long list of collaborators, which include both Canadian as well as international investigators, all who bring expertise to our study in terms of scientific experience and skills, and generally providing advice about not just the design of the study initially, but issues that we run into as we conduct the study. We also have an international scientific advisory board consisting of the following members, all who have quite a bit of experience collectively in cohort studies, larger studies, and again, in diverse scientific fields. We meet with these scientific advisors twice per year to get valuable advice about issues that have cropped up during the study. And it's an incredible resource because obviously there's no manual as to how to effectively run a large national cohort study. And so the expertise of these individuals is of great value to CanProCo. So overall, as I've alluded to multiple times already, the overarching scientific aim of CanProCo is to evaluate a wide spectrum of factors associated with clinical disease progression in MS. So both the onset of progression and rate of progression. So we're trying to look in detail at when progression starts and how quickly it happens in people who do have progression. And we want to assess how all of these factors relate to one another to influence progression in MS. So one of the really unique aspects of CanProCo, and Probably powerful aspects of CanProCo is the fact that we're collecting such a wide range of information on each of our CanProCo participants. And again, specifically, because we designed the study with the intention of trying to better understand progression, this is a really important point because this is how it differs from many existing cohort studies that are currently studying people living with MS around the world. So in many of our CanProCo participants, as many of you are well aware, not only do we collect clinical data, but we collect clinical MRIs that are done for clinical purposes, but also research-grade MRIs of the brain and spinal cord that use many novel sequences that will allow us to look in more detail at tissue structures in the brain and spinal cord. And then we collect blood. And if you get cerebral spinal fluid studies done for clinical purposes, we also collect cerebral spinal fluid. So really a wide range of factors collected. And we're, again, one of the most unique aspects of CanProCo is that we will be looking at how all of these factors influence progression in MS and not just kind of focusing tunnel vision on one specific factor. So that probably is one of the most powerful aspects of CanProCo, that we're really doing a deep dive of data collection, bringing in scientific experts in pretty diverse fields from around the country so that we can see how all of these factors interact to cause progression in MS. And the diagram here is just a schematic illustration of that. I should also emphasize that we're also collecting health administrative data in British Columbia and Alberta, which again is very powerful because Canada has a public healthcare system. We're able to collect data on larger scales like this. And so we'll be looking at, all the way from health systems factors, down to clinical factors, down to tissue factors, down to molecular factors that cause progression in MS, and seeing how all of these factors interact with one another. In addition to the primary objectives, we do have exploratory objectives. So the integrative objectives are just looking at how all of these various factors interact with one another to cause progression in MS. But the exploratory objectives, we are incorporating, iPad facilitated data collection, and then initially also tried to incorporate a smartphone app called Floodlight. And the goal of doing this is to explore how novel data collection and analysis strategies may improve the assessment of progression in people with MS. I alluded to earlier that one of the major clinical challenges that we have in the clinic when trying to examine people with progression or progressive subtypes of MS is that we don't yet have ideal tools to measure progression. And so this is an important exploratory objective. We're hopeful that we'll be able to eventually incorporate some sort of digital tool that will allow us to better monitor different types of progression in people with MS. So our study design, and many of you are familiar with this just because when you were providing consent to be in the study, you likely went over many of these study design aspects. So it is a 5-year cohort study, and it's prospective, meaning that patients who consent to the study are followed over time. As I alluded to earlier, there are 5 participating sites, which represent the largest MS clinics and academic centers across Canada, and it goes almost from coast to coast. And then we have very specific inclusion criteria, as you can see here, when we're enrolling people with relatively early relapsing remitting MS, relatively early primary progressive MS, as well as radiologically isolated syndrome, which is thought to be pre-symptomatic MS. And then we're including 1,000 patients total. But in a subset of 500 patients, we're including more detailed data collection, which includes not just a wealth of clinical data and clinical MRI collection, but also research grade MRIs of the brain and spinal cord, as well as biological samples. We're also including 50 healthy control participants. And this is what our study looks like. So this is a schematic just summarizing that at every clinical visit annually, You get quite a bit of clinical data collected. And then again, in the sub cohorts, which are about 500 people altogether, in addition to all of the clinical data and the clinical MRIs and the neurological exam, research MRIs, biological samples are collected as well. This is a slide just outlining that Floodlight digital smartphone app that I talked about earlier. Earlier in the study, we did try to incorporate this, but we found that the uptake was pretty poor, unfortunately. And then in people who had tried downloading the app, most people did not use it beyond a few weeks. So we know that there are newer, more refined versions of this app that are currently being developed, as well as other MS-specific apps. So hopefully in the next year, we'll be able to land on an app that if some participants are willing, we would be delighted for you to try using these apps, because again, this is one of the important exploratory aims of CanProCo, where we're trying to figure out if there are digital ways that will better allow us to monitor MS symptoms and progression. Many of you are familiar with this iPad device as well. This is used to collect data during your clinical visits and is looking at, the utility of using some of these digital devices to collect clinical information on people with MS, as well as administering specific tests that assess different aspects of neurological function. As many of you are aware of, we are also collecting a wide range of questionnaires looking at quality of life, fatigue, mood symptoms, work productivity. And then in BC and Alberta, we are linking to some of these provincial administrative databases, which will allow us to look at that data that are collected on a population level, which is also a major bonus of CanProCo, because in many places around the world, there is not a large kind of population level database like this that we can link data to. And then as we've already talked about, we're getting a lot of clinical data as well as research grade MRIs and biological samples in some of our sub-cohort participants. In terms of the primary outcomes, as expected, across the spectrum of MS, the outcome is really looking at the onset of progression and the rate of progression in different types of MS. And obviously in people with primary progressive MS, we do see disability progression over time. But even in people with relapsing remitting MS, we are also monitoring for disability progression. And because we know that some people have very mild MS and some people have more active MS, we don't necessarily expect to see progression in everybody. But this is the value of the study, because we are trying to evaluate why some people progress pretty rapidly while others do not. We do have a number of secondary outcomes as well. So the primary outcome of disability progression is measured using a global neurological disability scale called the Expanded Disability Status Score. But there are a number of secondary outcomes using some of these other clinical tests that we're collecting at study visits, many of them using the iPad device. So we're looking at a scale called the Multiple Sclerosis Functional Composite, looking at visual acuity. And then again, from an exploratory standpoint, if we are able to land on a digital app that many people are able to use, we may be able to even evaluate if we can see progression in some of these clinical tests that are administered by digital apps. So, I've alluded to many of these points a few times during the talk, but CanProCo is a really innovative effort for many reasons. First of all, it's one of the first national efforts that are directed at understanding progression in MS. Again, there are many cohort studies around the world, but none that have been designed specifically with this purpose in mind. So we're very hopeful that it will contribute to the field in this regard. It's a powerful study and innovative, again, because we collect data from very diverse scientific fields. So it's not just clinical data or imaging data, but we're collecting clinical, imaging, health administrative, as well as biological data. And so this will enable us to look at many different factors relevant to progression in MS. We also have the possibility for data linkages with existing large databases, including the Floodlight Open Study, MS Paths, MS Base. These are all existing registries or studies around the world where we have quite a bit of similarities in terms of data collection. CanProCo is an illustration of how a huge effort like this can be launched when multiple funding partners come together. And so this was obviously spearheaded by the MS Society of Canada, but federal funding agencies like Brain Canada and industry partners like Roche and Biogen, as well as the government of Alberta, have all come together to make this study happen. And then, last but not least, this is a uniquely Canadian effort. And so the eventual goal of all of this is obviously to try to better understand progression in MS, but ultimately what we want to do is take that understanding and create personalized care pathways for people living with MS, not just progressive MS, but every subtype of MS, so that we can provide optimal care and ultimately improve the quality of life of people living with MS in Canada and around the world. So in terms of progress to date, I've gone over many of these points, so forgive me if I go a little bit quickly, but the cohort officially launched in the spring of 2019, and I'm very proud to say that we're nearly at 90% recruitment, which is a huge accomplishment given that because of the pandemic, every site was actually on hold and not able to conduct any study activities for nearly 6 months. We have had a small proportion of people who have withdrawn to date, but considering that this is a very small proportion, I think it's safe to say that we're doing really well in terms of recruitment and all of the unexpected challenges related to COVID. This is just a table summarizing recruitment across our 5 study sites, and as you can see, each site is doing quite well. And then in terms of CanProCo productivity, we already have many presentations and publications that have been reported nationally and internationally. And so the CanProCo study itself, the international community is aware of it because we've presented at large international conferences. And then our protocol and rationale manuscript, which is a manuscript that's important to get out into the scientific domain, is currently under revision in BMC Neurology, which is an excellent journal. And we're hopeful that it'll be published in the next couple months. In terms of each of the scientific pillars, a lot of progress has been made, and I'm not going to steal the thunder of my colleagues and allow them to summarize all of the progress. But as you can see, quite a bit of scientific productivity. Larry Lind and Jacqueline Cragg were successful in obtaining a CIHR, which is a national funding agency project grant to support some additional analyses. And there have already been many publications and presentations at different scientific conferences and manuscripts that have been submitted for publication. Neuroimaging as well has made a huge amount of progress. And as you can see, already many presentations taking place at international conferences, and likely within the next 6 to 12 months, we will have a number of publications to show. And finally, neuroimmunology has been making quite a bit of progress with really novel technologies to assess different biological samples. And as you can see, again, a number of presentations have already taken place at national and international scientific conferences, and we are confident that we will have publications to show within the next 6 to 12 months. So that's where we're at. I hope I've been able to provide you with a general overview of CanProCo. In terms of future directions, we are really excited because we think recruitment will likely be complete either at the end of this year or early 2022. We were told at the beginning that, our timelines were pretty ambitious and, it might be difficult to recruit, but thanks to the interest of people like you, Despite the COVID pandemic, we've been incredibly successful with recruitment. We do have plans to integrate some people with pediatric onset MS who have been followed in a separate cohort for many years into CanProCo, likely within the next year. So you'll probably hear more about that next year. We have a number of potential new funding partners, and we're hoping that we can obtain more funding so that we can extend the duration of the cohort if at all possible. We're working on data access policies so that we can share CanProCo data with qualified investigators in Canada and from around the world, if appropriate, because I think it's very important in this information era to be able to share data with interested investigators around the world so that we are all learning and sharing from each other. And then, of course, this year we're going to be focusing on many of these preliminary analyses and publishing baseline data from our cohort so we can get it out there in the scientific community, which will contribute to the global community's understanding of progression in MS, which again is one of our greatest unmet clinical and scientific needs. So with that, I'd like to end and acknowledge the time and effort that each and every one of you have put forth to make this effort possible. And just on behalf of CanProCo, thank you for being willing to participate because without you, obviously, the study would not be possible at all. And I do believe that by working together, we can truly make a difference in the field and over time change the future of people living with MS. And then would also like to thank the CanProCo team. This is a huge effort that it's not just one person, but a whole group of investigators, clinicians, and research staff that make it possible. Our funders, which include the MS Society of Canada, Brain Canada, industry partners, and the province of Alberta, and the Canadian MS community as a whole. So thank you very much, and I'm open to any questions.
Great. Well, thank you so much, Dr. O, for providing an overview of the CanProCo study. Hi, everyone. My name is Camille. I'm a research specialist at the MS Society of Canada, and I will be moderating the Q&A with Dr. O today. It's also my pleasure to introduce Dr. Natalie Arper, who is a CanProCo co-investigator from the University of Montreal, and she has kindly offered to provide the French translation for today's Q&A session. So we will be using the next 15 minutes to answer questions submitted by event participants during the registration process. So I just wanted to remind our audience that you will have the opportunity to interact with CanProCo investigators and post a question live during the breakout sessions. If you have any additional questions, please save them for the breakout. So how this Q&A will work is that I will read a pre-submitted question from the audience in English. Dr. Arber will translate the question to French, and then Dr. O can provide an answer, which will again be translated to French. So if our speakers are ready, we can get started. So the first question that we received from the audience was, what were the biggest challenges the team faced when selecting the inclusion criteria and the different study groups for CanProCo?
Alors, bonjour tout le monde, je vais faire la traduction. Donc, quels ont été les plus grands défis auxquels l'équipe a été confrontée lors de la sélection des critères d'inclusion et des différents groupes inclus dans l'étude?
As you heard, it took a long time to land on the particular study design that we have right now. If we had unlimited funding, I think we would have wanted to include every person living with MS in Canada, but unfortunately, this was not the case. So we had to think really long and hard about what the best scientific design would be that would allow us in 5 years to look at all of the factors related to progression in MS. And so in the end, it was a struggle between how big or how small to make the cohort. And, it was very challenging at times because obviously we wanted to include as many different types of people living with MS as we could. But ultimately, these decisions were made based on what we thought would be the optimal patient populations that in 5 years, which is actually a pretty short amount of time in the grander scheme of MS, would allow us to evaluate various factors. And then it was an issue related to what budget we had. So again, if we had an unlimited budget, we would have likely wanted to include, tens of thousands of patients. But given that this was a study that had to last for 5 years, we had to land on a reasonable sample size that could allow us to understand things scientifically, but that also the budget would support.
OK, alors pour résumer les propos de Dr. O, donc comme vous avez pu voir dans le calendrier qu'elle a présenté, il y a eu de très longues discussions parmi tous les chercheurs qui sont impliqués dans l'étude, en collaboration aussi avec les examinateurs externes, et on ne pouvait pas inclure tous les patients. Ça aurait été notre souhait d'étudier un plus grand nombre de patients possible, mais malheureusement, on avait des contraintes budgétaires et aussi une contrainte de temps. Donc, lorsqu'on a choisi la taille des cohortes de patients, on s'est penché sérieusement sur quel type de patients pouvait nous donner le plus possible d'informations dans une période de 5 ans, parce qu'on savait qu'on avait un budget pour suivre les cohortes de 5 ans. Et on a choisi les tailles d'échantillons, de groupes de patients qu'on pense qui pourront nous donner le plus d'informations possible durant cette période, puis en allant chercher le plus d'informations possible dans toutes les sphères qu'on vous a mentionnées aujourd'hui.
All right. Now for the next question, an audience member also wanted to know what sorts of discussions took place among investigators before deciding which hospitals would be able to participate in this national study.
Alors, la question en français, c'était quel type de discussions ont eu lieu entre les chercheurs avant de décider quels hôpitaux seraient en mesure de participer à cette étude?
So another great question, and it was a difficult choice again, just because initially when the Canadian MS community was made aware of this opportunity, there was an contact made with every MS clinic in Canada, and Canada actually has over 30 different MS clinics in every province from coast to coast. And the vast majority of MS clinic directors around the country indicated that they wanted to participate. But again, it was a matter of, because, every site that participates, we need to make sure that it's adequately staffed with research staff so that, patients can be brought into the study site and data can be collected. And so unfortunately, we were not able to include every site that wanted to participate because of budgetary constraints. But in the end, we landed on sites that we thought were quite representative of the largest MS populations around the country. We did ensure that we had pretty good representation from most of the most populated provinces. And then some of the requirements were that, as you saw, we collect a lot of data from many different scientific fields in all participants. So all of the clinics that participated had to have an electronic database that, kind of organized all of their patients. They also had to have adequate facilities where they could get these research grade MRIs and collect the blood and spinal fluid samples in an adequate way so that they could be used and analyzed in CanProCo. So there were these bare minimum criteria that needed to be met. And then in the end, we just ensured that there was adequate representation around the country. And then, unfortunately, there were additional sites that could have participated, but because of the budget, we had to narrow it down to 5 sites.
Alors, c'était bien entendu des choix difficiles à faire parce que il y a eu beaucoup de contacts entre les différentes cliniques à travers le pays et la grande majorité des cliniques de sclérose en plaques à travers le Canada étaient prêtes à participer à cette étude-là. Mais malheureusement, on a dû faire des choix difficiles et en fait, les choix se sont basés sur les ressources qui étaient disponibles dans les différentes cliniques. Entre autres, il fallait que chacun des sites ait en place le personnel nécessaire pour récolter les données qu'on devait récolter. Et un des grands points aussi, il fallait aussi que ces cliniques aient des données électroniques. Donc, le fait que toutes les données accumulées des patients au cours des années étaient déjà dans un format électronique, ça pouvait faciliter la tâche d'implémenter cette étude-là dans ces cliniques-là. Un autre choix, un autre item qui est important, c'est qu'il fallait que les cliniques aient accès à des installations adéquates, par exemple au niveau de la résonance magnétique, que finalement les patients puissent aller passer les résonances magnétiques qui sont essentielles à chacune des années au cours des 5 ans. Et bien entendu, des groupes sur place qui étaient prêts à manipuler les échantillons de liquide céphalorachidien et de sang dans un délai très court pour justement les entreposer dans les meilleurs délais. Il y a aussi le fait qu'on a souhaité avoir une représentation à travers le pays. Donc, on croit qu'on a réussi à aller chercher les sites, les cliniques dans les provinces les plus populeuses à travers le pays. Et aussi, on pense qu'on a une représentation quand même assez grande, là, je dirais, d'un océan à l'autre dans le Canada. Et bien entendu, c'est un choix difficile, mais bon, il fallait le faire, puis aussi à cause de contraintes budgétaires, comme vous pouvez vous imaginer, d'avoir du personnel de recherche et clinique qui participent à ces études-là, ça requiert quand même des budgets. Donc, on a dû se limiter aux 5 sites, mais on pense quand même qu'on est représentatif de la population canadienne atteinte de sclérose en plaques.
All right, thank you, Dr. Arbour and Dr. O. So our next question is a three-part question on risk factors for MS progression. And Dr. Arbour, if you want me to repeat the question, I'd be happy to do so when you're doing the French translation. So the question is, what do we know so far about what causes MS and or MS progression? Are there certain populations or places in the world where MS numbers are higher and MS progression is more severe? And how have you integrated what we already know about risk factors for progression into the CanProCo data set?
So,
J'y vais, juste...
Oh, yes, that's right.
Que savons-nous à ce jour des causes de la sclérose en plaques et ou de la progression de la sclérose en plaques? Y a-t-il certaines populations ou certains endroits dans le monde où le nombre de patients est plus élevé et la progression de la sclérose en plaques est plus sévère? Et comment avez-vous intégré ce que nous savons déjà sur les facteurs de risque de progression dans l'ensemble des données CanProCo?
So a very challenging question to answer, but I think we can say that there has been much progress made in the field in the last 10 to 20 years, so that we are understanding more and more that it's really as to what causes MS and what causes progression. Like many different autoimmune diseases, ultimately it's a combination of various factors in the environment as well as genetics. And so because we're aware of all of this, obviously within the design of CanProCo, we've included a number of things that we know are relevant to developing MS and what causes MS disease progression over time. But because this is one of these things where the more you know, the more you realize you don't know. This is exactly the reason why CanProCo exists, because there are certain things that we know are relevant to risk of MS, including, say, living further from the equator, having lower vitamin D levels, having a family member who has already been affected by MS. So these are all things that we know. And because of that, we've with that those types of points within data collection in CanProCo. But there are probably 10 to 100-fold things that we don't know. And this is why we are trying to collect as much data as we can from both a clinical perspective, biological perspective, and imaging perspective, and even from a health administrative perspective, because it might not just be genes and environment, but many other things, including the influence of how far you live, say, from an MS clinic, that can affect the rate at which you develop progression. Things like access to various healthcare resources, additional things like, say, dietary factors or education or all of these things. And that's why we're doing such a kind of wide net in terms of how much data we want to collect, because we realize there's things we know, but there's a lot of things that we don't yet know.
C'est une question très difficile à répondre parce qu'il y a beaucoup de de facteurs qu'on connaît, beaucoup de facteurs qu'on ne connaît pas. Ce qui est très clair, c'est que la combinaison de facteurs environnementaux et de facteurs génétiques combinés ensemble vont créer un facteur de risque de développer la sclérose en plaques et vont aussi affecter le développement de la sclérose en plaques. Donc, on a de plus en plus de données. Malheureusement, il y a encore beaucoup de facteurs qui sont inconnus. Il y a des facteurs qu'on connaît, par exemple, la distance à laquelle on s'éloigne de l'équateur. On sait que le risque d'avoir la sclérose en plaques est plus grand, donc d'où le Canada a une très haute très grande prévalence de cette maladie-là. On connaît aussi de plus en plus mieux l'impact de la vitamine D, donc des déficiences en vitamine D pourraient être un facteur de risque. Et aussi, si vous avez un ou des membres de votre famille ou donc l'aspect génétique qui est affecté par la sclérose en plaques, votre risque de développer la maladie est bien entendu plus grand. Ce qu'on sait, il y a beaucoup d'autres facteurs sur lesquels on voudrait se pencher, puis on espère que notre étude pourra jeter peut-être un peu de lumière. Entre autres, certains facteurs qui nous intéressent, c'est est-ce que votre progression dans la maladie pourrait être influencée, par exemple, à quelle distance vous vivez d'une clinique de sclérose en plaques? Quels sont votre accès aux soins de santé? Est-ce que ça peut avoir un impact ou pas sur votre capacité à faire face à la maladie, puis aussi au développement de cette maladie-là. Le niveau d'éducation et aussi la diète, donc quel genre de style de vie finalement que les patients adoptent, est-ce que ça peut avoir un impact ou pas sur la progression? Donc, ce sont certaines des données que finalement, certains des facteurs qui pourraient avoir un impact sur la progression de la maladie qu'on aimerait, qu'on espère qu'on pourra en partie répondre. Mais bien entendu, c'est très complexe et on espère qu'en utilisant une grande cohorte sur plusieurs années, on va pouvoir arriver à certaines des réponses.
All right, thank you so much. That actually brings us to the end of our 15-minute Q&A session. I know it was brief and a lot of people has a lot of questions. We will try to answer your questions in the breakout sessions that will follow up. But I'd like again to thank Dr. O and Dr. Arbor for this live Q&A, and thank you everyone for your attention. We will now actually share a video recording from the representatives of each CanProCo scientific pillar. So momentarily, you will be hearing from Dr. Shannon Colin for the neuroimaging pillar. Dr. Alexander Pratt will talk about the neuroimmunology pillar, and then Dr. Tony Trabolsi will speak about the health outcomes and epidemiology pillar. So please bear with me as I share my.
Hi, my name is Shannon Coland. I'm a physicist based at UBC in Vancouver, Canada, and I'm very happy to be presenting about the neuroimaging in CanProCo on behalf of the whole CanProCo team, and in particular, my neuroimaging pillar co-leads, Drs. Ji-Won Oh and Roger Tam. This is my disclosure slide. So people living with MS are generally very familiar with MRI. It's really become critical to diagnosis in MS. When my grandfather was diagnosed back in the 60s, we didn't have MRI yet, so it was an even slower and more painful process than it is now. It's also important for monitoring how MS is changing over time, and it's really been instrumental to clinical trials for therapies because we can see changes happening on a shorter time scale than what we measure in clinic. And so this is an example of how things might change over time in one individual with MS. And you can see these bright spots or lesions, which are the scars that we see in the brain that change over time in MS. One of the reasons that MRI is really useful is that just by changing the settings on the scanner, we can get out different types of images that highlight different features that are happening in the brain. And so here's a few examples of what we would typically acquire in a clinical exam, what I call conventional MRI. And already you can see that you can make the lesions look very different using these different types of images and tell which ones are new and which ones have been around for a while. But that's not quite enough information. So the conventional MRI is very useful, but it is qualitative. So the actual brightness doesn't really mean anything. It's not very specific. So we can see that there's these bright spots, but we don't know exactly what the biology is that's changing that's causing it to be bright. We know that there's changes going on outside of those lesions, but we're not picking them up with this conventional exam. And it's not super related to how someone actually feels. And so these are two different individuals living with MS. You can see they have very different amounts of lesions, but they actually had the same clinical disability as we measured it. So this is why we want to move to also include non-conventional or advanced MRI, in our studies of MS so that we can get more specific to different pathologies, for example, myelin, which we know is damaged in MS, look at tissue that looks normal on our clinical MRI, and also do quantitative measurements, really look at those numbers between groups and over time. And this is how we think we're going to be able to better track disease progression and also measure whether therapies are working. So these are some of the different types of advanced MRI that we're able to acquire in CanProCo. So in addition to the clinical exams, we also get these research exams for our subgroups of people. And so here we have myelin motor imaging, which is specific to myelin in the brain, which we know is damaged in MS. And here we're showing more damage in the red and purple areas that we're not picking up on the conventional scans. MTR is also sensitive to myelin, and we usually use this to look at changes in different types of lesions. We can look at the cortical thickness, so how thick that layer of gray matter around the outside of the brain is. We can look at diffusion imaging, which tells us about how water is moving in the brain and how that changes with damage. Frequency shift imaging, we can do things like look at the veins in the middle of lesions and different types of lesions that might be more related to progression. And we can also look more at spinal cord, which is often missed in conventional clinical scans, but obviously really important to what's happening in MS. So these are examples of the actual images that we're acquiring in CanProCo. And so all these different types of images we're acquiring in the brain in addition to our normal clinical exam. And all these different images in the spinal cord. And it's really amazing that we can get all of this inside of an hour. It's a lot. And we're really grateful to our wonderful volunteers and our technologists. I want to very quickly show you some preliminary results. And these were put together by a medical student, Akash Chopra, during his training on a research rotation. And he was actually invited to present these results at an international MS conference. So obviously people are very excited and interested in this. So here he's looking at spinal cord data from our baseline scans in CanProCo and looking at how big the cross-section of the spinal cord is in our different groups. And so the progressive group in blue on the left has a smaller area than the relapsing remitting group in orange, the radiologically isolated syndrome or RIS in gray, and our controls without MS. But what's really driving this difference in the size of the cord? Well, we think myelin damage is a big part of it. And so he looked at our myelin measurement in the spinal cord, and sure enough, we see that that is lower in the progressive group. And now you can really see the steps between these different groups, with the more progressive groups seeing more of this myelin damage. So we feel like we're on the right track for looking at things that are going to relate to progression over time. So ultimately, we're going to build up models combining all of these different pillars and really get to the bottom of this. Really like to acknowledge especially our research participants, and of course also the families and carers, our funders, the clinical staff, our technologists, the entire CanProCo team and collaborators, and also our young trainees who are getting involved right from the beginning. So thank you very much.
Hello, my name is Alex Pratt. I'm a clinical scientist from Montreal. And we're going to be talking about CanProCo initiative for sure, and some of the biology pillar experiment that we have in this. So I'm going to show you two techniques, very exciting techniques that we're using to identify factors that might be involved in the severity of the disease and in the fact that some patients progress and others don't. So the objective of the biology pillar of CanProCo is to discover new biomarkers of MS progression using flow cytometry and single-cell RNA sequencing. And it's also to provide the Canadian MS community with very valuable, high-quality biological material matching with clinical and MRI data. So within the CanProCo cohort, we have several sub-cohorts. The first subcohort is what we call the radiologically isolated syndrome. The second subcohort is the classical untreated relapsing remitting multiple sclerosis. And then we have a smaller PP, primary progressive MS cohort. So we've done some preliminary experiments with selected controls and selected MS patient that we've subjected to flow cytometry and single-cell RNA sequencing to see whether we would be able to differentiate the blood from the control from the blood from a MS patient in a completely unbiased way. So you can see on this graphic that these population of cells that are located in the blood of MS patients are extremely different from these population of cells that are found in the blood of healthy donors. And one of this population is called the CD8 T lymphocyte. The CD8 T lymphocytes are very aggressive lymphocytes that fight against infection, but also against cancer. And in the blood of MS patient, you can see that there's an increase in the number of these CD8 T lymphocytes when compared to healthy donor. So the question is the second technique that we're using matching the flow cytometry? So is single cell RNA sequencing matching the FACS data? Because most of CanProCo will be to perform single cell RNA sequencing of blood leukocytes, white blood cells in MS patient. So this is a diagram which shows by single cell RNA-seq all the cells that MS patients have in their body, in their blood. And if you overlay that of the control with the MS patient together, you can see that there are different population. In pink here, different population of cells that are found in the blood of MS patient as compared to control. So this population, or this population, or even this population is present in higher number in the blood of MS patient than in control. And two of these populations are, per se, CD8 lymphocytes and CD4 lymphocytes. So these CD8 lymphocytes that were identified by flow cytometry initially are also identified by single-cell RNA-seq. So in conclusion, we believe that combining flow cytometry and single-cell RNA-seq, as we do in CanProCo, will easily and reliably identify different cell types and provide quantitative information about their relative frequency in MS, and we'll be able to match that with the trajectory of the disease over the course of 6 or 7 years. So we'll know in 7 years which patient had what type of blood cells that are associated with a better or a worse prognosis. And using 3 MS patient and 3 healthy control in an unbiased computer-based analysis of both single-cell RNA-seq and FACS data, we identify unbalanced immune cell population in MS patient. So this is the goal of the biology pillar of CanProCo. The goal is to identify blood markers that will allow us to be able to predict which patient will do better, which patient will do worse, so that we can actually adapt the treatment depending on the profile of the patient. So I'd like to thank my team in Montreal, as well as the Multiple Sclerosis Society for funding this CanProCo initiative. Thank you.
Hello, I'm Tony Trabolsi, a neurologist at the University of British Columbia in Vancouver. Along with my co-chairs of the epidemiology pillar, with Larry Lind, also professor at the University of British Columbia, and Scott Pitton, a professor in Calgary, we've assembled a brilliant team of young researchers who will help us redefine multiple sclerosis. Our team has considerable experience in epidemiology, pharmacology, psychiatry, and economics. And with this team and the data we'll be collecting, we'll be able to better understand the impact of multiple sclerosis on patients, families, and society using a variety of new measurements that haven't been used before in the field. We also want to examine how other features, such as underlying health conditions, might impact on a person's future with MS. And then combine the state-of-the-art research with neuroimmunology and neuroimaging to really develop a personalized treatment approach for the current generation of people with MS and future generations. Our feeling is that the experience of multiple sclerosis just can't be summarized alone at the cellular level. We want to know how it impacts on individuals and their families. Certainly as a clinician, I've been in practice for about 20 years, I can understand how MS can affect everyone quite differently. And I see that our current way of monitoring the impact of MS on people is only touching the surface and misses subtle or obvious challenges such as fatigue, depression, memory, employment, and relationships. The second thing I notice is that MS is very different today than it was 20 years ago, and I truly hope that's mostly in a good way. However, a lot of the decisions we're making are based on information that's perhaps out of date. And traditional research methods and studies have really relied on simple measures of functional ability, the neurologic examination. And this really only identifies the tip of the iceberg and doesn't take into account many other critical symptoms and the impact of MS. If we make decisions on the neurologic exam alone, we might be missing important changes that are occurring. And I've certainly seen that in my own practice, where someone's examination seems stable over time, but clearly they're losing function in other ways. The value of all the sophisticated immunology and MRI data being collected in CanProCo needs to relate to how MS affects individuals, and for that we need to collect information that is meaningful to patients. One of our goals is to capture those important details that have meaning to individuals with MS and to better understand MS today and in the future. It'll be valuable to integrate all this new knowledge with the latest imaging and immunology technology. And that approach is really what we call big data science, using artificial intelligence or machine learning to combine complex data to find patterns that we might not see otherwise. We believe that this approach will redefine how we think about, understand, and care for MS for the next 10 to 20 years. And as we analyze all this information through big data science, we'll have at least what we hope 2 major impacts. We hope it'll lead to the basis of personalized medicine, meaning that each individual with MS is unique, and by comparing their unique features to the larger comprehensive model created from the CanProCo data, we may be better able to determine an individual's risk for future disability and treat with the best therapies that match with their profile. We hope that this approach to personalized medicine will translate into long-term best health. The other advantage of this wealth of data is to use this new knowledge to better inform government, insurers, employers, and families as to what resources are required to decrease disability and the impact of disability for individuals and for society. We look forward to your questions and around this very exciting project. Thank you.